Hello students and welcome to Legacy IAS Academy. So like every other year, this year also the Nobel Prizes are being given, and in today's session we will be discussing the Nobel Prize in Medicine, which is given for the discovery of microRNA and its importance in gene regulation. So we will see what this is all about. What's the news? Mr. Rukun, along with his collaborator, Mr. Vikram Ambrose from the University of Massachusetts, have received this Nobel Prize for the discovery of, remember, direct question in prelims could be asked, for the discovery of microRNA or miRNA. Now, one important thing is to not confuse it with mRNA, which is the messenger RNA. I hope this is absolutely clear. Now, what does this discovery do? So to understand that, let us understand more about this particular discovery. So they have discovered microRNA. Now, what is this microRNA? It is a new class of tiny. That is why they are named micro, because they are small, tiny. It is a new class which has been discovered now, new class of tiny RNA molecules which play an important role in the regulation of genes, and this role which they play in the regulation of genes is very important for the development of multicellular organisms, including human beings. So this microRNA has been discovered in animals which are multicellular, and it not just plays the role in multicellular animal but also human beings, who itself is a multicellular organism, right? So that is the importance. Now, because of this discovery, what is it going to revolutionize? It is going to revolutionize the way we look at the things related to genes, how they are controlled and how they are expressed in living organisms. So that is the importance it holds. We will get into the detail of it even further. So what happens is, in our cells, they replicate, they multiply. At times they replicate so much that it can cause cancer. So not just in regulation of genes, but it is going to also help in, probably in future, identifying the cure for cancer as well. We'll see how that is done. So these are the importance. Now, since we are talking about miRNA, there are other concepts related to RNA, or you can say the genetic makeup. So what are they? First, it's DNA, then there is RNA. So we will first look at these two things, then we will look at messenger RNA, and then finally we will look at the newly discovered microRNA. And when we look at all these four things, we will completely understand the development of protein molecules in the body and the development of a particular trait in a human being. At the same time, how it's going to help in curing cancer in the future, which is a possibility. So let's start with the difference between, or the thing about, DNA and RNA. So this is DNA and this is RNA. Now what is the difference between the two? Na means nucleic acid. So Na is here, both have nucleic acid. The only difference is, this is ribose nucleic acid, whereas this is deoxyribose nucleic acid. Deoxyribose nucleic acid, whereas this is ribose nucleic acid. So that is why, since it's just ribose, this also has ribose. Why is the word dioxy? It has two strands. Can you see this? This is one sorry, this is one strand and this is another strand. What about this? This is a single strand only. That is why ribose, and that is why this is deoxyribose. Now, they are present independently on their own. Remember this differentiation. Whereas ribose nucleic acid are synthesized from the DNA as and when they are required. So simply, there are two strands. These two strands can detach and form what? Single strand. I hope that is clear. It is as simple as oxygen having two molecules. You bifurcate them, so it becomes nascent oxygen nascent oxygen. However, this nascent oxygen is very unstable and it may combine again. That instability is not present here. So DNA is present on its own, whereas ribose nucleic acid, that is RNA, is synthesized from DNA as and when required. Right? So the only difference is the two different strands. Rest everything, the components inside them will remain the same. What do they contain? Cytosine, guanine, adenine and thymine. Here also cytosine, guanine, adenine and uracil. One change. Why this one change? Because it's a chemical. So since it's a chemical, when you split it, it does not necessarily split like a physical element. So if I break this particular thing, or if I break the body into two, the components remain the same, the chemicals remain the same. But if you look at the minute strands which are breaking away, that may change the complete, what do you call, the constituent of this. So the one change that happens is this. Remember, all of it is same. It is just a synthesis from DNA. Everything remains same. One thing changes from thymine to uracil. Right? And even that change is not much. The only change there is the change in the hydrocarbon which is getting detached. That's all that is happening. But because of that, there is a massive change. So this is the difference between RNA and DNA. Now, there is another concept of mRNA. The full form of mRNA is messenger RNA. So what does this messenger RNA do is simply it carries the message. By message, what we mean, this coding. Can you see this coding? Can you see this coding? This coding is carried by the messenger RNA. So what messenger RNA does? From the DNA, it takes this coding, attaches this coding. See, this coding is getting attached over here, and this coding gets attached with the help of mRNA, and it then helps in forming the protein from the DNA. So protein from the DNA can only be formed with the help of mRNA, which transfers the coding for the formation of protein. Consider this as your Swiggy or Zomato delivery which is happening. Getting the food, right? In the same manner, the coding is being transferred or given in the formation of protein. That is why the messenger. This is the message, the code. The code. This is literally called the code. The code is the message, and the carrier is mRNA, that is the messenger RNA. So now we know the DNA, we know RNA, we know messenger RNA and its role. Now let us understand what is microRNA that has been discovered and what is its importance. Now, to simply understand this, what will happen is, let's come back to this picture. What will happen is, messenger RNA is carrying the information, carrying the code, carrying the message. When it is carrying the message, it is leading to the formation of protein, right? Now, this is going to basically define the growth, development, everything of a human being. How the genes is there, how your protein is developed, leads to the growth and development. Now, that growth and development, what happens is, let's say you formed a particular personality. There would be some fine tuning that would be required. Imagine like this, you create a painting, you make a painting. So you make a holistic big painting, you color it, but you might be required to add certain fine details into that painting, right? With a micro level of brush, with a subtle touch of the brush. Now that fine tuning, so basically that fine tuning is basically done by miRNA or microRNA. MicroRNA does the fine tuning. Not just that, it ensures which RNA gets converted into protein, which does not. It ensures which should be allowed, which should be not. That is the importance. And this discovery can lead to solution for or cure for cancer. How? Because in cancer, what happens? Multiplication, right? If we can have a miRNA which decides, no, this multiplication should not be allowed, it is hampering, then we can actually mimic this miRNA in order to ensure this multiplication is stopped. So this is a possibility, not yet done, but opens a wide scope or potential for that. So what is the benefit or importance of miRNA, that is microRNA? It is the fine tuning which this microRNA does. Okay, so let us see. It's a small non-coding RNA. Non-coding RNA. What do you mean by non-coding? This microRNA will not consist of this. These are codes. So microRNA will not consist of this. And these codes are made up of what? Cytosine, guanine, adenine and thymine. Cytosine, guanine, adenine and uracil. So these codes are not present. So microRNA will not be having these codes. So they are small, non-coding and helps in the regulation and expression of the genes. Now, unlike messenger RNA does what? It carries the instructions to make proteins. So messenger RNA is just a carrier, carries instructions to make proteins. MicroRNA, what it does, it binds to a specific kind of RNA molecule and it helps in fine tuning, also preventing them from being translated into proteins. It can stop them, it can allow them, it can fine tune them, or by degrading the mRNA together. So it can prevent them from being translated into protein. So there is this, let's say there is this RNA. Now the RNA can get converted into protein with the help of mRNA, right? But microRNA can prevent this from happening. This is the strength of microRNA. And also it can allow it. Since it can prevent it, it can also allow it. So it is the one that is deciding which RNA can be converted, should be converted to protein, which RNA should not be or cannot be or will not be converted to protein. I hope that is absolutely clear. So this is how it is. So what happens is, this information from the DNA is being broken and taken up by the mRNA, and mRNA is formed. Now what microRNA will do, it does not want this to be converted into protein. So it attaches itself to the mRNA and ensures that this is not converted into protein. I hope that's absolutely clear. Okay. Now, importance, as we saw, it will help, it of course fine tunes the various cellular processes like development, growth, metabolism, etc. It's essential in maintaining the cell, the multicellular organism as well as the function of the cell. Okay. Now, this discovery will have huge implication on our understanding of cellular differentiation. Cellular differentiation means breaking up of the cell, or development means development of the cell, and disease processes, how disease arise and how they can be cured. I hope that's clear. So this Nobel Prize research has opened up a lot of further fields of research in the field of curing of cancer, in the field of not just cancer, various other diseases also, as we can see, cancer, diabetes and autoimmune disorders. Autoimmune disorders are what? These are disorders which are you can say having on which your immune system does not have any impact. See, any disease, when it happens, we have an immune system to it, body responds, reacts to it. Autoimmune diseases are so dangerous because when they happen, your body is not responding only. Nothing doing. No response. Even when people had COVID, the body was reacting, responding. You know, when you get some, let's say some fever, viral, this, that, so not fever, but any viral infection, your body's temperature rises. That is nothing but fight. The body is fighting that viral, right? Now in autoimmune diseases, the body completely withdraws. It does not fight, and you have to completely rely on the medications, surgery, etc., etc. Right? So this discovery will advance our basically our potential or our effort to ensure the cure for cancer, diabetes, as well as autoimmune disorders. and hence it is very important. So all the things that could be asked for prelims, it's clear. I hope you have understood how the process entirely works. So even if in mains a specific static question on mRNA, miRNA, DNA, RNA is asked, you should be able to answer. Thank you so much.